Today, we're going to take a look at a new computational method. It's one that's designed to model a particularly challenging class of proteins, specifically those that don't have a single fixed structure. This research really extends the capabilities of deep learning tools into a new frontier of structural biology.
So over the last few years, deep learning methods like AlphaFold have just completely transformed structural biology. They're able to predict the three-dimensional shapes of folded proteins with an accuracy that's right up there with high-resolution experimental techniques. And that's allowed us to model hundreds of millions of protein structures.
But all this success raised a really important question. How do these tools handle proteins that aren't static? You see, a lot of proteins are inherently dynamic and flexible, and they don't just fold into one stable shape. This presents a completely different kind of prediction problem.
To really dig into this, we first have to understand the specific challenge that protein disorder presents.
These proteins are known as intrinsically disordered proteins, or IDPs. Now, unlike their folded cousins, IDPs exist as a collection of structures that are constantly changing. This is a concept called a structural ensemble, and for them, their function often comes directly from this flexibility. So the distinction here is fundamental. A folded protein has one main structure, and that structure dictates what it does. A disordered protein, on the other hand, is this dynamic group of structures. Because of that, standard prediction tools that are looking for a single answer often report low confidence for these regions, simply because there's no single correct structure to find.
In practice, this means when you use standard AlphaFold on a protein with disordered regions, its own confidence score, the PLDDT score, is usually low for those sections. This is the model's way of telling you it can't lock down a single well-defined structure, which, in a way, accurately reflects the protein's disordered nature.
So, to solve this problem, researchers have developed a new computational approach that uses AlphaFold's outputs, but in a totally different way.
The method is called AlphaFold-Metainference. Instead of trying to find one single structure, it takes the distances between amino acid pairs that AlphaFold predicts. These distances then act as guides, or restraints, inside computer simulations that model how atoms and molecules move. The goal isn't to get one structure, but an entire ensemble of them.
Now, the key insight that makes all of this possible is a very specific observation. While AlphaFold can't give you a static structure for a disordered protein, its prediction of the average distances between the amino acids is still highly accurate. This distance information is pulled from an AlphaFold output called a distogram.
So the workflow can be broken down into 4 steps. First, you use AlphaFold's distogram to predict those average distances. Second, you use those distances as flexible restraints in a simulation. Third, you run molecular dynamic simulations to explore all the possible protein shapes that fit those restraints. And finally, you collect all those shapes to build the full structural ensemble.
A new computational method always needs rigorous validation, so the researchers compared the structural ensembles their method generated against actual experimental data. This data came from a technique called small-angle X-ray scattering, or SAXS, which is used to figure out the overall shape and size of proteins in a solution.
Okay, let's look at a specific case study, the TDP43 protein. This one is associated with the neurodegenerative disease ALS. The standard AlphaFold prediction for TDP43 shows some well-defined folded parts, but also these long, disordered regions where its prediction confidence is very low. In contrast, the AlphaFold-Metainference method doesn't give you a single shape. Instead, it generates a whole collection of different conformations. And this ensemble really captures the known flexibility of the protein's disordered regions, which gives us a much more physically realistic picture.
We can actually quantify this improvement using a metric called the Kullback-Leibler divergence, or DKL. You can compare the models to the experimental SAXS data. A lower DKL value means a better match with reality. And as you can see, the AlphaFold-Metainference ensemble is a much closer match than the standard single-structure prediction.
And this result wasn't just a one-off for TDP43. The method was tested on a whole set of 6 different proteins that have both folded and disordered parts. And in every single case, the ensembles from AlphaFold-Metainference showed significantly better agreement with experimental data, compared to the single structures from standard AlphaFold.
This kind of advancement has direct implications for understanding human health, mainly because many proteins involved in diseases are themselves intrinsically disordered.
The study successfully applied this new method to several of these proteins. This includes TDP43, which is involved in ALS, Ataxin-3, which is linked to Machado-Joseph disease,
and the human prion protein responsible for Creutzfeldt-Jakob disease. Having accurate structural models for these is a very important step toward understanding how they work and what happens when they don't.
So this work opens up the possibility of modeling the complete structural and dynamic character of many important proteins for the very first time. And this leads us to a final, forward-looking question. If we can see not just a static snapshot, but the full range of motion of disease-related proteins, how might that change our fundamental approach to designing drugs and therapies that interact with them?